Okay, let's pick up where we left off. We had done the karyotype and the idiogram, no? Now we'll look at how they are similar and how they are different. The karyotyping technique gives both karyotype and the idiogram. Both show the total number of a cell. I think a total number of chromosomes in a cell. It's more like that. They show the morphological features of the chromosome. And in both pictures, the chromosomes are ordered in a series of decreasing size. Okay, so that is what is similar about them. This is total number of chromosomes in a cell. Okay. Then let's see how they are different. A karyotype is it shows us the actual picture of the chromosome set of a cell. Let me highlight this keyword, chromosome set. Okay, so the karyotype shows us the exact picture, the exact chromosome set of the cell when we arrange them in the homologous pairs in a series of decreasing size. Okay, so the exact set of chromosome when we arrange them in pairs. Okay. Idiogram is the diagrammatic representation of the karyotype of an individual. Okay, so karyotype you get the set, and then idiogram you get the diagram. Okay, the diagrammatic representation. So this is the key difference between karyotype and idiogram. When comparing karyotype and idiogram in terms of identification of regions within chromosomes, it is easy to identify regions within chromosome in an idiogram rather than a karyotype, because remember, in idiogram you are getting bands of the genes, no? So you know exactly which gene is where. Okay, going to the uses of the karyotype. The karyotypes of, let's see what's written here, of different groups are sometimes compared, and similarities in karyotypes are presumed to present evolutionary relationships. Okay, so what this point means is that, let's say in this field, not field matlab microscopic field, in this micro photograph, this is the karyotype of, let's say, human. Then this is the karyotype of, let's say, a chimpanzee. Then this one is of orangutan, you know. Then this one is of your macaw, round tail macaw or something, that other monkey. Then this one is your baboon, no. So by studying the difference in these, the karyotypes, we can have a picture of how man evolved or how the orangutan evolved, from the differences. Suppose, just an example, okay, this is not scientifically backed. I have not read a paper or something. I'm just saying, just suppose the karyotype of a human is very similar to a baboon, okay? Just a guesswork. And it is not as similar to, it is a little bit more similar to orangutan, very less similar to chimpanzee, and the least similar to a macaw. So we can just conclude that maybe we have evolved from the baboon, no? And then the baboon evolved from the orangutan. Okay, so you can conclude, you can compare the similarities, and you can deduce how they evolved. Okay, then point number 2, it also suggests primitive or advanced features of an organism. Okay, the karyotype showing large differences between smallest and largest chromosome of the set having few metacentric chromosome is called as a symmetric karyotype, which is considered to be of advanced feature, when compared to the symmetric karyotype, which has all metacentric chromosomes of the same size. Okay, so what this says is that, like human, no, our karyotype of human, I had already shown you the diagram, no, we have chromosome 1 longest, then 2, then 3, then 4, on and so forth, till these very short chromosomes, and you have the 23, which is X and Y. Okay, we have the most differences, we have highest and lowest. Okay, so we consider ourselves as the most evolved, okay, as compared to, let's say, maybe this organism, which has, for example, 1 2 3 4 5 6 7 8. Let's say this organism has 8 chromosomes, and all of them are more or less of the same type, so we consider it to be less advanced. Okay, because they are all of the same shape, they are more or less symmetrical. Okay, so that I think it point 2 is clear. Then looking at the material of the chromosome, the material of the chromosome is the chromatin. Okay, so a chromosome is made up of chromatin. Depending on their staining properties with the basic dyes, especially the name of the dye that we stain is Feulgen reagent, we can identify 2 types of chromatin during the interphase nucleus. Okay, so what is the chromosome made up of? Then you will tell me, Miss, chromosome is made up of chromatin. Okay, then chromatin has 2 types. One is the euchromatin, one is the heterochromatin. So let's see what they are. Okay, now the euchromatin, let's say this is your chromosome. Okay, then you are seeing some light bands, some dark bands, some lightly stained bands, then some very darkly stained bands. Similarly on the other arm, so these are dark. Okay, dark. So this is dark and these are lightly stained. This is lightly stained. Okay, so this lightly stained part, okay, like this or this, the white part, the lightly stained part, this one is your euchromatin. Okay, because they stain lightly because they are only partially condensed. Remember I told you, suppose this is a scarf. Okay, so you squeeze this scarf, you squeeze it, it becomes very condensed. So such a long scarf you can fit in just this much space. Okay, so some parts are highly condensed, some parts are less condensed. Okay, some part is highly condensed, some part are less condensed. So this less condensed part, this is your euchromatin. Okay, so let me read what they've written. Portion of the chromosome that stain lightly are only partially condensed. This chromatin is termed as euchromatin. Okay, so it represents most of the chromatin that disperse after mitosis has been completed. So euchromatin contains structural genes which replicate and transcribe during G1 and S phase of the interphase. It is considered genetically active chromatin since it has a role in the phenotype expression of the genes. In euchromatin, DNA is found to be packed in 3 to 8 nanometer fiber. So basically what it says is that the part that is slightly condensed as compared to this highly condensed part, this part is what is turned on. Okay, this part of the chromosome, this is highly condensed. The less condensed part, this is the active part. Okay, means this part of the gene, the lightly condensed one, it goes through your DNA replication, then transcription. Okay, so let's say this one, this is your DNA, then it will unwind, right? Then replication will happen. Then we are getting 2 strands of DNA, 2 homologous strands of DNA. So replication has happened. Then what happens? It will get transcribed also, no? So we'll get your rRNA. Okay, so the part which takes part, okay, during replication and transcription, okay, is your euchromatin part. Okay, let me repeat. I'll try to simplify it. Remember, I used to tell you about the junk DNA, the part we don't need. So that part will be in your heterochromatin part, okay, the very condensed one. So the lightly condensed one will take part in DNA replication, okay. It's take part in DNA replication, and then transcription, translation, all those, and protein synthesis. So everything is going on, and it happens during the G1 and the S phase of the interphase, because that is when your replication happens, during G phase and S phase. So whether you look like your mother or your father or your grandma or your grandpa, just remember that it is your euchromatin, okay. This is what is responsible for your inheritance, okay. It plays part in how you look like on the outside, okay. So I'll stop here for today. As this is a Saturday class, I'm also having not much energy. So I hope it's clear. Any doubt, please ask me. You can text me there in the WhatsApp.